so far in this first half is that this first half is all about generalizations about microbes. General aspects of how a cell works, how metabolism works, etc. And so all of these things, again, most microbes do something in the way we've described. Obviously, there's exceptions to every rule that we've given. But as these generalizations go, these are, in general, how microbes work. And so what we want to do today for our review for our midterm coming out on Thursday is we're going to take all these features and kind of tie them together. And you'll see how, even though we talk about them as individual aspects of microbiology, they're all really directly related as one cell function. So what we're going to do is see how all the different aspects we've talked about work together to function well. Front table will be fine. Okay. So the way we're going to do this is we're going to start by building a cell, and what you'll see is that as we build a cell, we're going to then fill in all the other gaps from the other sections that we've done. Right, so everybody who's doing this, come on up. Alright, so let's build a cell. So if we're going to build a cell, we need to start typically with the boundary of the cell, or cell membrane. And so how do we build cell membranes in biology?
Peptidoglycan.
So phospholipids.
Oh yeah,
Phospholipid bilayer. And in cartoon form, we typically draw it with these little heads and tails in this bilayer structure. And so what is the power of one of these phospholipids? Why are these good membranes? What are the parts of a phospholipid?
Hydrophilic and
So you have a hydrophilic, hydrophobic part. And so what are the three different parts of a phospholipid? Three different molecules that make it up. There's a central 3-carbon sugar. Anyone remember what that's called?
not at the moment.
That is glycerol. Now attached to 2 of those carbons are these long tails. So what are these called? Fatty acids. Right, so that's your hydrophobic part. And then attached to the 3rd carbon is the phosphate group. So that's where the phospho comes from. And we know that a phosphate group typically has a charge to it. And so because you have this charge here, this creates a hydrophilic portion. Because you have a fatty acids here, that creates a hydrophobic portion. And so as the tail portions tuck themselves into the middle of a bilayer structure to protect themselves from water, this kind of bilayer will naturally form because of the chemistry of these molecules. And so, in just about all of biology, a phospholipid bilayer is how we build a membrane. But in this class, we have to say in just about all of biology, because there is one exception to that. And so what was that exception? Archaea, and specifically high temperature archaea. And why? What happens if you take a fat and put it at high temperature? Melts apart. And so, obviously for a cell, it can't have its membrane melt apart. And so what kind of modification do we see in high temperature archaea?
Monolayer?
We have a monolayer instead. And kind of the easiest way to understand this or imagine this is basically the fatty acids are just going to link all the way through, thereby creating a monolayer. And that monolayer then becomes much more stable at high temperature. So that is the membrane of our cell. What are some of the other parts then that we need to fill in our cell? So let's build a bacterial cell. Cell wall, probably the defining characteristic of a bacterial cell. So what are some of the functions of the cell wall? Primarily it's a protective layer, but it does a few other things like provides the shape of the cell, so it determines what the shape or structure of the cell is. Anything else? Provides trigger pressure for the cell, so the cell doesn't explode. And also then it creates that periplasmic space between the membrane and the cell wall. Now in bacteria there are 2 kinds of cell walls, so what are those 2 kinds of cell walls?
Gram-positive, negative.
Gram-positive, gram-negative. If you were to describe the parts of a gram-positive cell wall in words, what is contained in a gram-positive cell wall?
Thick
peptidoglycan layer. And there are a few other things like TA, that teichoic acid, LTA, lipoteichoic acid, some proteins and stuff. But really, the peptidoglycan molecule is the defining molecule of the gram-positive side. How about on the gram-negative side? How would you describe that in your words? So you still have peptidoglycan, but it is now a thin layer.
It's 2 pre-carbonic spaces.
And then you have a second membrane, called the outer membrane. And that one has a molecule called LPS, lipopolysaccharide. And then there's some like pores and proteins and stuff. But really, this molecule is the defining molecule of the gram-negative side. And so, as we look at the structure of these two molecules, how would you describe the structure of peptidoglycan? Long strands of sugars, and how are those strands connected? By short peptides. And so, as we build our long strand of sugars, what are the two sugars that make my long strand? Nag and NAM. And where do these sugars come from? They're actually derivatives of glucose. And how are they connected? Beta 1,4 linkage. So what does that beta 1,4 mean?
Connects at the 1 and the 4.
So the 1,4 tells you connects the 1 and 4 carbons. And what does the beta part mean?
That's what you told us.
It's a beta sugar. Right? So beta sugar connected at 1 and 4. And so as we build these out and make a long chain of these, that creates our long strand of sugar. But now I want to link them through these short peptide chains. And so what are the 4 amino acids in my short peptide? L-ala, D-glu, M-DAP or L-lyse,
and D-ala.
and then D-ala. So first off, there's these L and D forms. What do those terms mean? They're the isomer forms, which essentially means they're the left and right-handed version of these amino acids. And which one of those is normal for amino acids? L or D? L. So normally amino acids are found in the L form. And so bacteria are actively changing a couple of these over into the D form. And then in this third position, you can either have this MDAP molecule or lysine. What kind of bacteria have each, and what is the significance of that? So what happens if you have MDAP in this third position versus what happens if you have lysine in this third position? So why is that important for the cell? Direct versus indirect crossbridge. So if you have MDAP, that is a direct crossbridge. And as we mentioned, all gram-negatives and then about half the gram-positives use MDAP. Lysine, on the other hand, requires an indirect crossbridge. And then the other half of the gram-positives use lysine. But essentially, as I take this building block and I repeat it millions of times, that is what creates the gram-positive cell wall. On the other hand, if I look at LPS, what is the defining characteristics, or what are the parts of LPS? So you have a lipid A portion. And that's the part that actually sticks into the membrane. Above that? Core polysaccharide. And what does that do? Or what is that? So polysaccharide is simply sugar. And then above that, the O antigen, or sometimes called the outer polysaccharide. And so as we define these parts, again, lipid A is a part that sticks into the membrane. And so we're going to have two sugars here with a phosphate group on each side, and then fatty acids. And how many do we need?
5.
At least 5. Above that, the core polysaccharide is a black box of sugars. And what are the minimum requirements for the core polysaccharide?
2 KDO.
2 KDO sugars. And then above that is the O antigen, and why, or what is that made up of?
Species-specific
So
sugar.
it's a species-specific sugar. And then it's repeated 30 to 40 times. And because it's species specific, that's what gives rise to this term antigen. So what does an antigen mean, and why is that important?
Antigen activates the adaptive immune response.
So because the LPS molecule, or the O antigen specifically, is species specific, we can use it to activate an adaptive immune response, and so you make antibodies against that, and that's one of the ways that we fight off infections. Good. What's another molecule that we use to build? So let's continue building our bacterial cell here. What else do we need in our cell?
Nucleoid.
Nucleoid, right? So every cell needs a nucleoid. And why? What is a nucleoid made up of?
Genetic information.
The genetic information for the cell, which means what is it made up of? DNA. And what is the function of DNA in a cell?
Construction.
Instructions for
Making
making proteins. What is the instructions for making a single protein called? A gene. And so that gene will be found in your DNA. But just a gene would not be enough to actually make a protein. There are some additional features that you need around that gene in order to actually turn it into a protein. So what else would you need around that gene? You know where it starts. You need a promoter. What else?
Terminator.
You know where it stops. You need a terminator. What else?
DNA polymer
You
enzyme.
need the enzyme to actually make the RNA. So you need RNA polymerase. And then
A TS.
that little TF, which allows you to
Turn activate.
activate a specific gene. What's the most common TF that's used by bacteria? Sigma.
70.
70. And as you have those four things in, you can actually turn those into a segment of RNA. Now in the RNA, you essentially need the four equivalent things to actually make your protein. So what are the four things that you need in the RNA?
Start codon.
You know where it starts. You need a start codon. And the start codon is always
AUG.
AUG, which codes for what amino acid?
My.
Methionine.